Yeah, good morning, everyone. We will get started. We are about 6 minutes beyond 9 o'clock, and hope you had a good day, half a day session yesterday with a lot of physiology and some historical background of interventional cardiology, mostly in the stent era. So today we will start with a presentation by certain members of the industry. And the first one is James Hickley from UK, and he represents Abbott, and he's going to give us a scientific presentation as to where Abbott is and where Abbott is headed. Go ahead, James.
Morning, everyone. I hope you can hear me in the room.
Yes, we can.
Thank you. Thank you very much for the opportunity to speak with you today. My name is James Hickey. As mentioned there, I work for Abbott as an R&D manager, and my team and I are responsible for the development of DES. I'm currently in Ireland. I'm currently sitting in the southeast of Ireland. It's a town called Kilkenny, close to the plant where we house our R&D group, which is called Clonmel. And that is where we currently develop drug-eluting stents and balloon dilatation catheters. Okay. Okay. So I would like to speak to you today on our experience in the development of DES devices, if I may be so bold. And so I will just begin. And to discuss the development of DES, I'm going to focus on Abbott's device sciences. That is what my experience is. And I can go through that. I suppose just a little about our design approach when developing these DES. I suppose it can be described as safety and performance without compromise. And when I reflect on this design approach, a couple of comments spring to mind. In developing DES, we always prioritize safety without compromise. All of the designs, they do involve choices and balancing between a number of factors. And it's the package as a whole that delivers its safety and performance. We, in the design lab, pay a lot of attention to the things that you actually see in your lab, but also the things you don't see. The clinical outcome is a function of the full design package, and the Xience family progression from Xience V to the most modern stent systems will continuously raise the bar on safety and true procedural success. Okay, so just to speak a little bit about Xience, obviously. It was designed as an overall system, with each component contributing to its excellent performance. So the Xience system is comprised of a thin strut cobalt chromium 3-link multi-link design. A multi-layered coating utilizes a primer and a fluorinated copolymer. An everolimus drug with a low dose and controlled release through the restenosis cascade, and an advanced delivery system tuned to transmit force efficiently to deliver the stent to its intended target. So I am going to take each of these components in turn and discuss its features, its development effort, and challenges. And I will take questions at the end. So let's just talk about the stent, okay? So it's true to say that stents are being used in ever more challenging anatomy and clinical situations. This has fueled continued stent design development to meet these demands. The development of the stent itself requires a large effort, a large amount of resource and time to complete. From the initial design concept to ready to use in the clinical setting, typically tens of thousands of stents are produced in the development cycle and tested. It's an iterative process, and many iterations of stent design are manufactured and tested to ensure that program goals are met with respect to performance and safety. To help us with the design, modern design tools are utilized. For example, 3D drawing packages, finite element analysis, OCT guided modeling, etc. A large battery of mechanical benchtop tests are also completed on candidate designs. Typically, we test them to failure, to understand the design completely in order to produce a safe and effective device. These benchtop tests range from, but are not limited to, radial force resistance testing, stent stiffness, stent recoil, end ring stability, fatigue fracture to 10 million cycles, testing in many different orientations, deployment accuracy of the stent, stent dislodgement force, and also its compatibility, of course, with MRI, corrosion resistance, and the list goes on and on. I could name many, many more tests that we do. Suffice it to say that we would completely characterize the device before we allow it to go forward for clinical testing. In effect, much effort is expended in designing the stent platform that prioritizes the safety and performance of the device without compromise. I suppose the result of this design effort in Abbott is the Xience family of stents. And if I focus on some of the characteristics of the Xience stent, these would be as follows. It's obviously a cobalt chrome material, which is widely used, which uses an alloy of cobalt, chromium, along with tungsten and nickel and a small amount of iron. This has allowed for greater strength characteristics over stainless steel, but also reducing strut thickness to optimize metal to artery ratios. The Xience stent pattern is made up of repeating rings connected together from peak to valley with 3 nonlinear links, which deliver a great amount of longitudinal strength, which is key to resisting the compressive and elongation forces that may be applied to a stent during the intervention procedures. The cell design is repeated along the length of the stent. The more rings you have, the longer the stent is. But within those rings, the cell design remains the same. It is repeated it's predictable along the length of the stent, and when in combination with a non-linear link, the design ensures a predictable scaffold integrity and stent stability upon deployment. Furthermore, the stent design delivers uniform scaffolding for even drug distribution, vessel support, and minimal plaque prolapse, which are all key factors in vivo. Over the years, the post-dilation diameter has been increased to allow for greater flexibility when implanting into tapered vessels. The size matrix has also increased to include up to the 48mm stent length, and nominal diameters have gone from 2 and now up to 4.5 and 5.0 in the latest large vessel devices to treat longer diffuse lesions and maybe more proximal lesions. Okay. So now I would like to discuss the Xience stent fluoropolymer. So this is one of the unique features of the Xience and one of its major development activities within the design project for DES. The selection of the stent coating is a key factor. Coating options typically range from a no polymer option to the selection of a durable or a bioresorbable coating. So the Abbott development for Xience focused on a durable fluoropolymer, and I will discuss its developments and advantages now and why we selected it. So the Xience coating is made up of a primer layer and a polymer which houses the everolimus drug. The Xience permanent fluoropolymer, fluorinated polymer, was a deliberate choice after an extensive screening assessment over many years, taking in over 70 alternatives. Extensive research and development led to the adoption of a primer layer material, PBMA, poly N-butyl methacrylate, and a PVDF HFP, polyvinyl iodine fluoride cohexafluoropropylene, or fluoropolymer for short. Also, the primer allows for adhesion of the polymer to the stent surface, and it's encapsulated completely underneath the polymer. PVDF and HFP is a highly fluorinated inert polymer utilized to modulate drug release. This polymer is blended with everolimus to give a highly miscible solution that can be sprayed onto the metal stent platforms in a uniform manner. And that is how we apply the drug we spray it on. The modulation of the drug release can be tuned by varying the ratio of the VDF and HFP portion, as this controls the thermomechanical properties to deliver optimum drug release rate. Within the polymer, strong covalent carbon-carbon and carbon fluoride bonds exist, and these provide outstanding chemical stability in vivo. The carbon fluoride bond is actually the strongest in organic chemistry. So the result of all this is a coating with very desirable characteristics of toughness, flexibility, and elasticity for usage with a stent. So upon selection of the coating materials, the extensive development of the coating application to the stent was undertaken. This involved years of development and study to ensure very precise application of the coating to tightly control the drug dosage, elution rates, and coating integrity. So what is the benefit of all this extensive chemistry and process development? So many in vivo benchtop studies have been completed on the Xience coating. It was found and reported in literature that the fluorinated polymer coating induces selective absorption of albumin protein competitively over fibrinogen. This protein modulation of albumin subsequently minimizes both platelet adhesion, activation, and leukocyte recruitment. And the suppression of platelet adhesion is an important factor in stent thrombosis and is a result of the surface chemistry of the fluoropolymer. From preclinical in vivo testing, the evidence suggests that fluoropassivation and thromboresistance, evidence supports the fluoropassivation and thromboresistance nature of the fluoropolymer, which combined supports the overall safety profile of the Xience drug-eluting stent. So in conclusion, the Xience permanent fluoropolymer was a deliberate choice after an extensive screening assessment of over 70 alternatives. It offers long-term durability and stability to the implant and supports the overall safety profile of the Xience drug-eluting stent. And it was a major undertaking by the company to develop that coating and process around it. So I'm going to talk a little bit about the everolimus. Not a lot. As you know, it's a widely used drug in the space, and it is utilized to limit neointimal hyperplasia. I suppose the feature of the everolimus therapeutic drug is that it's optimized for the restenosis reduction. As I spoke about already, coupling it with the polymer, the Xience elution rate is matched to the restenosis cascade. It has a low dose of 100 micrograms per centimeter squared and a broad therapeutic range. Okay. So the everolimus typically is a market leading, which is a proven broadly used therapeutic drug. So now I'm going to come to the delivery system. And this is the last feature that I will speak about. Over the years, the delivery systems have undergone a transformation driven by the need to treat lesions in more complex situations. Much development effort has been exerted to improve the catheter shaft performance over the years. To discuss a few, every DS program places a large emphasis on stent deliverability and invests much time and effort in prototyping, test model development, the testing of the devices, and implementing improvements. This drives balloon and shaft development to find a balance between flexibility and pushability. Modern devices exhibiting kink-resistant shafts along with thin balloon designs with ultra-low crossing profiles have been developed. This typically results in systems with low crossing forces and excellent pushability. Another key attribute is stent security, while maintaining the deliverability performance. Actually, in my experience, stent security on the balloon is by far the attribute that requires the largest volume of development units to be manufactured and tested and drives development timelines. It's typically the longest studies and the largest studies that you will complete. This attribute is a key focus for the Xience device. The device utilizes balloon pillowing technology and advanced crimping technology to ensure that the stent, which has been developed over many Xience years, is completely retained on the balloon until you decide when to deploy it into its location. The ability to accurately deploy the stent is also a key focus area. Many stent designs and stent placements on balloons are currently available. Xience utilizes a stent edge on the balloon marker design so that the position of the stent in the crimp state is known, and coupling this with the predictable Xience stent scaffold results in accurate and predictable stent deployment to the target lesion. Each size within the matrix will have a bespoke design to ensure that the stent, balloon, and marker separation distance are specified to deliver the accurate and predictable stent deployment. Another area of focus for manufacturers of DES in recent years has been to increase the dimensional envelope that the DES devices operate in. We see this in increased post-dilatation expansion ranges and expansion of size matrices offering to accommodate tapered vessels and to treat more proximal and longer diffuse lesions. Okay. So in conclusion, lots of effort to improve the cath lab experience has been completed over the years. In closing, Xience is an extensively studied, widely DES with over 20 million devices implanted. I would like to iterate that Xience was designed as an overall system, with each component contributing to its excellent performance. It was designed with an ethos of safety and performance without compromise, and it has been my privilege to work on this program and to be able to impact positively people's lives over the last couple of years. So I'd like to thank you for the opportunity of speaking with you today. I will take any questions, and I hope you have a great conference today as well. Okay, thank you.
Okay, so now it's time for discussion, questions, comments. I think that everybody have quite a good experience with Xience stent. Practically, we started with Xience was one of the first stents after pairing of Cypher and Taxus. Ravi?
Can you comment on the safety of your stent in patients that receive abbreviated DAPT? I mean, what's the characteristic of the stent that allows one to stop the DAPT either in 28, the Xience 2890 data?
Sure. Thank you for the question. It's a great question. The characteristics of the Xience that allow that is manifold. It starts with the stent having the low metal to artery ratio. Okay, so the contact of the stent with the actual intima media layers is at a low ratio. But I suppose the main feature that promotes the safety of the device in this instance is its fluorinated polymer. That's what's doing this. As I spoke in the talk there, the polymer's chemistry actually interacts with the body to selectively bind albumin. And once to fluoropassivate, it's called fluoropassivating the stent surface, so that thrombogenic materials don't stick to the stent. And it allows for the endothelium to heal efficiently and completely. And that, I think, is the main reason why the DAPT regimes and the studies and the times that the studies support have been so successful. It's down to the chemistry of the polymer, which is durable on the Xience device. It stays there. It always stays there to protect the stent.
Thank you.
So that means that the polymer promotes endothelization faster.
It promotes that it promotes that the surface of the stent doesn't gather thrombogenic agents while the endothelium goes about its business of healing itself. That's typically how it works, and the short DAPT trials and clinical studies have shown the effectiveness of that when it comes to stent thrombosis.
Okay. So, other questions or comments? If not, okay. So thank you so much.
My pleasure.
It was a really nice presentation, and we will go further.
Thank you very much.
Thank you so much.
Bye. Have a great day.
Thank you. So the next one is a presentation for Biotronic. Hello.
Hello.
And Jens Martin. So now we will see the newest technology from Biotronic. Okay.
Yeah, good morning to the panel. Good morning to the auditorium. It's a pleasure to be here in Sofia. And yeah, I take the opportunity to give you an overview for the Biotronic technology. First of all, I start with the drug-eluting stent, and I thought it's good to have an overview, and if you talk about improvements, it also should be proved in high-quality studies. So also I brought just a little bit of update regarding the clinical data setup. So in 2021, I saw this picture during EuroPCR, and I think it's a very interesting picture because it shows where we are today. My mission for this morning is to let you have an update on the ultra-thin strutted drug-eluting stents, but we see in Europe a strong trend in the use of DCBs, and maybe there's a new start with metallic bioabsorbable scaffolds, and this is what I want to take you a little bit to a journey regarding these technologies. I think we can say today, and also I show you an editorial picture from Antonio Colombo as a conclusion afterwards. We see a strong trend. I think most of us know the picture, know about the minimizing of the stent struts. And you see on the left corner down there also the effect on the blood flow. And the thinner the strut, the earlier, if the strut is well opposed to the vessel wall, we know there are less turbulences, less risk of thrombogenicity. And overall, we end up with 60 microns. One company is even offering a smaller one, going down to 55, but we decided to stay with 60 microns because it's also about post-dilatation options. And very interesting, if you see the 3 ones ultra-thin, so below 70 microns, ultra-thin strutted DES, you see 3 systems, the Orzero, the Supraflex Cruise, and the Coroflex ESA Neo from B. Braun. And if you see who contributed with real RCT data and data you can trust in, well published, you see that the Orzero, let's say, has absolutely the lead regarding clinical trial data for ultra-thin strutted DES. Just to let you know, today you can buy different DES, more than 20 different models still, and you see there are lots of ideas how to bring the drug onto the stent. We decided to have a hybrid coating, so a passive coating combined with an active coating. That means we don't need a primer for the polymer, because we also learned, for example, times of Nobori drug-eluting stents, that also a primer as a base, how to fix the polymer onto the stent, can be a topic. And we were all surprised that the Cypher was beating out the Nobori at that time, and that's one of the reasons why Biotronic decided to have this hybrid approach. You see it here very well shown that we have a resorbable polymer. It's very important to understand we have good reasons why we have a very slow degradable polymer, ultra-thin PLLA, where the sirolimus is in, because the slower the degradation, the lower the amount of inflammation and the better the characteristics for healing. This is also directly linked to the opportunities you have with short dub therapy, and you see here in rainbow colors, this is the passively coated backbone. This is our prokinetic energy, which is then coded, and as an Orzero Mission or the Syncero Pro, we sell it as a modern drug-eluting stent. If you look, there are different approaches. The colleague of Abbott showed their science concept very well, and here you see the overview who is still using also passive coatings. And as I said, not only in coronary but also in peripheral, we have very good reasons to have our ProBio. This is amorphous silicon carbide, and as I said, with this we don't need a primer and can directly coat it with a PLLA. We are here at the BCC Congress and also talking about bifurcations, and I think everybody knows what counts and what is important for modern drug-eluting stents, also for the usage of complex bifurcations and real bifurcation treatment. You see here the overview, but still there's a lack of clinical data, and we have from the Netherlands, from Clemens van Birgelen, we do have the BioResort trial. This was interesting because comparing the Orzero versus Synergy and versus the older Medtronic drug-eluting stent, and I think also the outcome from BioResort is interesting. Here we have some detailed pictures. What did we do in research and development? The stent is performing pretty well, and we designed a new delivery system, a new balloon catheter, and of course the design is very important, and we do have a dual helix design with our stent. What is very beneficial also here, we see that the helically or the sinusoidal stent designs may be beneficial. You see it as a group of my O0 Mission and the Resolute Onyx representing the sinusoidal design, and the double helix is the design of the O0 Synzero stent. Here we do have some fresh pictures and direct comparisons, and you can see there is a good improvement overall in delivery systems. The marker designs are nice, and the stents are well mounted or brought to the balloon and the delivery system, but still there are differences if you look to the details, how the balloon is folded, how does the start of the balloon shoulder looks like, and there are still differences. And I think for today, if we talk about ultra-thin strutted DES like O0 Mission, it's very important to understand that a deep embedding into the balloon is very important, not to afraid to lose the stent during tricky, complex interventions. And you can see there's a straight line on the right side for the O0 Mission, that we don't have any balloon material disturbing, and that we have a deep embedded stent into the balloon material. And of course, crossability, more and more we are facing very heavily calcified lesion. Crossability is one of the main features we are looking for. And of course, internally in Biotronic, the old O0 was the bench, and you can see that with the O0 Mission, with the new system, we were even able to improve. What is also very important to understand, there are in between we have some more models ultra thin, but it's very important to understand there are differences regarding the geometry of the stent. And if you have, let's say, 60 by 60, it's a totally difference compared to our system, that we have a strut thickness of ultra thin 60, but the strut width with 75 microns is bigger. And this ratio counts, and there is a theory from radial strength calculation. You see on the right side, there are big difference. For example, if you directly compare the O0 Mission and the Supraflex Cruise, for example. These are pictures from the Eastern Sea, from the north of Germany, taken by Professor Oehner. And this is how a stent in REM looks like if the balloon was used up to rated burst pressure. And you can see here for the O0, this is how a modern coating looks like, and it looks pretty much better and different to the old and first generation DES. Regarding bifurcation treatment, especially on the right side, if you talk about DK crush, I think at that area where we built a new metallic neocarina, this is very interesting. How does the surface coating behave? And everybody of us knows if you crush two drug-eluting stents which are surface coated, you will peel away a part of the active coating. And this is the reason why we stay with a passive coating behind. And this makes a really pretty good biocompatibility also for that metallic carina area. As I said, Antonio Colombo, I think, summed this up in a very nice way in his editorial in Neurointervention. And he says, of course, if you talk about ultra-thin strutted DES, the deliverability is much better. We have less thrombogenicity. We have a better vascular healing, a faster endothelialization regarding the smaller footprint we leave. And there is just one area where ultra-thin strutted stents today have to face a limitation. This is if you talk about left main treatment and ostial lesions, because there's a much higher radial force coming from the wall. I think more and more, I said clinical data are important, and I think the relevance of registries is growing. So, for example, from Sweden, the SCAR registry gives us a very nice picture also regarding long-term data, and there you can see that the O0 is within a group of very nice performing DES, modern DES, where you can say there is a group what seems to be favorable over a long-term follow-up in this national registry. If you look, let's say, and I think this is an interesting publication also from Eurointervention Journal, which drug-eluting stent also contributed with data in all-comers RCTs. There are not too many, and there is Orzero, one of the biggest suppliers for these data. And we, I think, are the only company at the moment, also with long-term follow-up, who had, let's say, the idea to compare directly to a very well acting drug-eluting stent, well-known, the science. And here we have not only the FDA approval study, but also the BioSTEMI study done in Switzerland. And here for the BioSTEMI study, it was interesting comparing the Orzero versus the science, because above 3 millimeter in diameter, more or less, they have the same strut thickness. So it's not only ultra-thin strutted stents, but also in this group, above 3 millimeter in diameter, we saw differences between the performance of the stents, and there is where the strut thickness is really, really comparable. And here we have the data which were shown at the last Congress last year, and with a very nice result showing there are differences in the DES concepts, and this is continued up to a long-term follow-up. And I think these were data, and Biotronik was strong enough to go for a superiority study where you can see there are, let's say, not big differences, but measurable differences, even if you compare an ultra-thin strutted DES versus a well-performing, well-known science drug-eluting stent. We also touched some minutes ago the short DUB topic. So there are some companies who feel strong enough to do a direct RCT study, not comparing clinical data versus a historical group. Here we felt strong enough also to pick up the Resolute Onyx, a well-performing drug-eluting stent, what we also know from the Dutch study from the BioResort trial, what I mentioned before. And here we have the direct comparison, around 2,000 patients, Orzero Mission versus Resolute Onyx. And we know that the Resolute Onyx is a well-performing DES, having the CE mark for 1 month short DUB. And I just show the primary outcome at 1 year. And you can see, and that's interesting, the calculation was the expected rates were above 9% for the primary endpoint. This is a combined endpoint, as you can see here. And we estimated or calculated 9.4%, and both drug-eluting stents performed really, really well with a very low event rate for the primary outcome after 1 year, and this is really remarkable. And this shows that today the modern DES perform in a very, very nice way, and they are safe and effective, and this is also published in circulation. As I said, if you compare drug-eluting stents, the question is there a new gold standard? And as I said, ultra-thin is not ultra-thin. So we have to be careful. If we compare ultra-thin DES, it's very interesting to have a closer look, and this was presented at EuroPCR, for example, Orzero versus Coroflex ESA. We do have different coding concepts. We have here a combination for B. Brown, Sirolimus, and Propofol. And the interesting thing was for this third generation DES that we have statistically significantly different outcomes even after 1 year, knowing that the polymer takes 2 years for degradation for Orzero. And here you can see both stents are ultra-thin strutted, but after 1 year there's a significant benefit for the Orzero. And if you have a combined endpoint like NACE, so including also major bleedings, you have a significant difference, a positive effect for the Orzero in this direct comparison. So the last 4 minutes I want to use also to focus on the bioabsorbable scaffolds. I would love to talk also about DCBs, because I think there are a lot of things going on at the moment. The rationale for the use of a resorbable stent, and I'm concentrating here on the magnesium scaffolds, is that we can have flatter curves after the time of resorption. So hopefully these graphs you see on the right side for the modern drug-eluting stents, these curves can be flatter if we talk about next generation scaffolds. And the first parameter, the resorption kinetic, is for us also key, combined with the alloy, so the resorption should be as fast as possible that the patient can really benefit from this. And I think there are also very interesting publications in the last year. And I come from Freiburg, so Franz Josef Neumann was working next to me in Bad Krotzing. And this was very interesting because he's a more conservative guy, always bringing interesting discussion points why he is still skeptical regarding the magnesium scaffolds. But you see in the lower part of the picture his comment in this publication. He says, the leave nothing behind concept is a very attractive concept, and we see first positive clinical data for magnesium. Or like his friend Adnan Kastrati commented, we do have hope for the magnesium technology. And this makes me very proud. And there is a history. When I started working with Biotronic in 1997, 1 year after, we started the AMS project. So since that time, we are working on the topic. So the name for the new product is still the study device name because we don't have CE mark. We expect this for the next month. So the last official name is not mentioned here, but in the studies we talk about last generation. This is DREAMS 3G. And we have, again, a resorbable magnesium scaffold. The abbreviation we are using is RMS. And here we do have a backbone with a new alloy, and this is made by Biotronic just for these scaffold technologies. And it's very important to understand the differences between the new alloy, the BioMag, and the old alloy. And Michael Joner from the German Heart Center in Munich had a very nice publication last year, also in Neurointervention, comparing the metallic scaffolds. And you see on the right side the picture where you see the degradation process, magnesium versus iron, and the iron takes 5 to 6 years of resorption, and the magnesium is just taking 1 year of resorption. And that's a fascinating point, that we have a very biocompatible material and a fast resorption process. And that means we do have with this concept a clear benefit for the patient, because after 1 year more or less it's completely degraded and the patient benefit comes in an early time frame. And here we do have, some of you do have experiences with the Magmaris. You see in the lower part on the left side, the picture B shows the picture for the preclinical testings for the Magmaris, and above are the testing results for the new magnesium scaffold, the DREAMS 3G. And here you can see the white arrows are the areas where the degradation process led to that point that the connection was lost, like part of the degradation process. And you can see the number of arrows is much lower with the new one, with the DREAMS 3G. And that means stability is much better, much longer. And we are now in a time frame more than 3 months, up to 4 months of mechanical stability, having this as a metallic implant. And here in this picture, you can see the new marker concept. On the left side, we have very thin struts for the magnesium scaffold. We go lower than 100 microns for the small vessel size, and we have a new alloy, a combination of magnesium and aluminium with a really low amount of aluminium, and this makes a very good performance regarding comparison also for mechanical characteristics to the older version. So the new version has a much better radial force, for example. With respect to the time, I skip. I just want to refer to Professor Haude, our CCI from Germany, to our first-in-human study, BioMag 1. And I just want to share one last picture, and this explains very well the difference between drug-eluting stents and a resorbable magnesium scaffold, because this concept means the struts are visible, for example, at 6 months follow-up, but after 6 months, the resorption leads to that fact that you don't have a metallic implant anymore. So the shadows disappear regarding OCT, and at 12 months you have a completely, more or less completely resorbed scaffold, and this was proven, and this is also published by Professor Yona and Dr. Segushi from Japan, that we have a resorption degree of almost 100%. And this is my last picture, and this is what you don't see with any other scaffold in the world, that at 6 months, we call it the Irish Hills, you just have, let's say, at 7 o'clock area, you just have, let's say, one hint where the struts were, but at 12 months you can identify the area where the struts were. And I think this is a really innovative new concept, and this makes the BioMag, the Biotronic new alloy, and hopefully we get CE mark in the middle of February, and then we will launch the product, I think, in March, and also starting the big RCT versus the science drug-eluting stand in April. This is the plan, and this is my update. Thank you.
Thank you.
Thank you. I have a little bit, let's say, a little bit more modestly enthusiastic about the bioresorbable scaffold, because I implanted the first 2 Magmaris stents in Bulgaria a couple of years ago. And when we made an 1-year IVUS at that time, during the control, practically the stent was still visible in IVUS. I sent the records to Biotronic, and they told me that the magnesium was replaced from calcium, and but more or less the stent was visibly there. That was the very first Magmaris, and don't look at me so bad. The guys from Biotronic probably are angry on me in this moment, but that was the truth, and they know that, because I gave them the CDs from the IVUS, and that was surprising, because the statement was that there would be nothing behind, but there was. At 12 months there was. So this is my initial experience with Magmaris. It will be interesting, the new generation stent, how it will perform. I would like to ask Gianluca regarding the flow dynamics, because you perform a lot of tests and you publish a lot of studies practically with Marco Zun. And
first of all, thanks to refer to my study, because I didn't suspect that someone find them of any importance, so thank you so much. On the contrary, I think that we speak a lot of time, we spoke all the time about the characteristic of the stent, the design of the strut, and whatever. I think that one very important thing is to refer to rheology of the vessel and to minimize stent strut, as not only because the interaction with the vessel wall is better, but is because if you take the strut of low profile and you use ultra thin strut, you maintain the rheology of the vessel in a more physiological way. And beyond the implantation technique in complex situation, and we found with computational flow dynamics that some implantation techniques are better or others in terms of wall shear stress force in the segment covered by the stent. I think that the novelty of, I was one of the very first users of Biotronic in Italy at the start of the experience with Biotronic, when the Biotronic stent was the first one to be really ultra thin. The other was, were 19 microns, no? And the novelty that I think that the company didn't think at that time is that the rheology of the vessel is maintained much better with an ultra thin stent strut than with other kind of larger strut. So I think that it's important to recognize that there is not only the polymer, the design, whatever, is that you have to maintain the flow within the vessel with a healthy features. Healthy features means wall shear stress forces not low, not too high, in the mean. Thank you.
So, may
Yes, yes,
Yeah,
sure.
just I have to comment
Yeah.
on your remark. It's important because there's a big difference between all other polymeric or tyrocore systems and the magnesium scaffold. What you told us, like with intravascular imaging, you can follow the resorption process. And the difference is we leave something behind. It's not like with PLA, so we don't have Krebs cycles here where we leave, let's say, more or less nothing. Here, we leave something. And that's the difference. So, and with the old IVUS, also with virtual histology, we were able to follow the resorption process. And it's part of the concept that you see the calcium apatite, and it's the same for the new one, the Freesolve. You see magnesium phosphate and aluminium phosphate at the end of the resorption process. So this is part of the concept, and you cannot compare pictures, what we know from absorb, dissolve, whatever, with this magnesium technology. And this is very important to understand. You cannot compare the metallic approach to the polymeric approach, because this is a different concept, and the benefit here is that we have a very biocompatible resorption product. This is a very good, a lot of water content, and this makes what we leave behind biocompatible and very flexible. So we are not stiffening the vessel. And this is just what I have to say, because that's part of the concept that you see the resorbed area.
Okay, maybe you should change the
Can I ask you sort of
the name because of
sorry,
the resorbable. Okay.
Dominic, can I ask
Yes,
you,
1%.
I would like actually to ask this question to all of you that you are presenting today. I understand that being better than the best is a very hard task, and we have best products today. How much sort of decided the industry is to invest on this? Are you going to invest in this? You think that's a main thing that you have to invest?
Yeah.
Are you decided?
Yeah,
And
like, like I said, so we will start the RCT trial, but we all know that the differences between drug-eluting stents today and the difference of a modern scaffold versus a modern drug-eluting stent, it's really tough to show superiority, but we are brave enough to start in April the RCT trial, 2,000 patients, the new DREAMS 3G or the new magnesium scaffold versus the science. And we are brave enough to do so, because this means this is a very expensive RCT trial, and this means our first step to show there's also partially superiority. But the tricky thing is, let's say with 2,000 patients, to show this as a first step randomized control trial.
And what is going to be the focus? Bioabsorption?
Yes, and the point is that after 12 months, that the trigger from a permanent implant has changed to a resorption product, what is biocompatible and what will not cause additional events. And that, like I said, after 1 to 2 years, that we can flatten the curves regarding the event rates like target lesion failure, and that also the reasons, for example, there's no fear of having neoatherosclerosis or to have late triggers coming from a permanent implant. How big the differences will be, we don't know, and we know if you use heavily modern imaging that the results are close to perfect. So this is really a challenge for us then also to show significant benefits.
So Biotronik is dedicated and is dedicated to absorption and thinks that is going to resolve the long duct and all the other problems that possibly we will still have with the stents.
Yeah.
Thank you. That's very important.
What do you think that it will be more challenging, the thrombosis, restenosis, or TLR?
Yeah. If you
So,
see,
you know, in this point, like
Yeah, yeah.
getting, knowing how the stand or the platform performs, what do you think it will be more challenging of these three things?
so I think if we have, we always say if you implant a scaffold, the procedure needs a little bit more time and love, so to say. You have to prepare the lesion close to perfect. And then you will see a very good result. But in real world, we know sometimes time is missing for a perfect lesion preparation. And the good thing is what we see, and this is also published now, that the difference between the new and the old magnesium scaffold is the new one is a cooler guy. So the new magnesium scaffold doesn't care about the plug characteristics. With the Magmaris, we had some cases where the support was too short, and we had some TLR cases after 1.5 to 2 months where we said, why does this happen? And this is a big difference with the new magnesium scaffold. We had an OCT study looking for what kind of plug do we see and how does the support look like. And with the new magnesium scaffold, this is also recently published, the new one doesn't take care how does the plug look like. And this gives us strengths that also in a real world setting that the performance might be good.
Thank you.
Okay, so let's go further. The next speaker is me.
Oh, it's you. Okay. So the next speaker is very well known to us, Dobry Vasilev. Are you representing NESMAR, is it? Or are
No,
you
this is Boston Scientific.
So it's a more of a scientific presentation, so you can introduce the topic and yourself if you like.
And the topic will be some products of the stents of Boston, the evolution of Boston Scientific stents, and the new Megatron stent characteristics, mechanics, and applicability in left main stenting and right osteo stenting, which are quite a challenging vocations for treatment. Имаме ли образ? Actually, I like to concentrate at the beginning in the evolution of the treatment of coronary artery disease, which started with balloon angioplasty, first performed from Andreas Grünzig in 1977, and practically Grünzig made the first device in his kitchen, and later on he applied it to human. It is interesting that the first patient, the result from the first patient was very durable. I saw this patient in 2008. He was invited during the TCT meeting, and it was quite interesting. And that was actually a left main case. But with the balloon angioplasty, there is a vessel dissection, there is a elastic recoil, which was the main problem and caused around up to 15 to 20% of complications, and acute vessel closure was actually the most severe of this one. Probably the older operators remember that in Bulgaria, if we wanted to do a PTCA on the proximal LAD, there was a need for surgical standby to be done. And in 1986, Jacques Puel with Ulrich Zigvart, after complicated life case in one old lady with exactly acute vessel closure of the proximal LAD, they implanted the first bare metal stent. The first bare metal stent was created from Julio Palmas and Richard Schatz, and for this one in 2008 at 20th anniversary of TCT. Julio Palmas has he showed all the history of how this stent appeared, but later on we and yesterday Fouvar showed that as there was a problem with the restenosis, with the intimal hyperplasia, which caused around 20 to 30% of restenosis with the bare metal stents, and then appeared drug-eluting stents. And Boston Scientific actually was one of the first companies with introducing the drug-eluting stents. As you know, the Cypher was the first approved this, and then appeared Taxus just 2 years after the results from Raveo, and this is why we practically in 2004 that we had two drug-eluting stents in Europe, and it was a huge improvement for the treatment of coronary artery disease. The evolution of next stents, next drug-eluting stents from Boston was quite astonishing. In 2006, Boston Scientific bought Guidon, and then in 2010, it appeared the Promus stent. It was distributed parallelly with the Xience. That was the same stent practically. And but later on, the Boston developed their own technology, and in 2013 to 2014, the Synergy stent appeared, and it was one of the first with biodegradable polymer. And now we have Synergy, the last modification of Synergy, with which is one of the best drug-eluting stents which we have. This is one of my workhorse stents, which I use almost every day, practically. And in 2016, the stent was approved for the shorter DAPT therapy, and in 2019, there was a labeling that the stent is approved for using in high bleeding risk patient, and in 2020 appeared Megatron. Megatron was appeared as a specially dedicated stent for the left main for the proximal large vessel disease. Practically, Taxus and Synergy much less, but Taxus was mainly criticized because of the two connectors and a little bit, let's say, there was a problem with the axial force and longitudinal shortening of the stent. Later on, there was a addition of the third connector between stent struts, and the axial strength was improved. Why we need a dedicated stent for the left main? Just because this is a location where the anatomy is specific. The wall of the left main is a continuation of the aorta, and there is a lot of elastic fibers there, a lot of elastic fibers, almost missing muscle fibers, and there is no adventitia at that region. So this makes the left main prone to elastic recoil, and there is a need for really dedicated stent for this region. Moreover, that there are a lot of procedural problems with the stenting of left main.
with not always good visualization of all segments, proximal, mid, and distal of the distal bifurcation of the left main, and some problems with the guide catheter engagement, and the similar procedural problems are existing in right coronary. However, the right coronary ostium is completely different from the left main ostium. There is a muscle band at the ostium, at the proximal part, and this is why there is a so frequent spasm after catheter engagement at that area. And this is why it is so important to give nitroglycerin, to be very gentle when you engage the right coronary artery. This area is prone to spasm, as I said, and there is a specific different shapes of the ostium, and there is a different shape of the occurrence of the artery from the aorta. So practically these are two locations very similar on the first side, but actually very different, but with similar requirements. The other point is because of the better results when we have higher minimal stent area, but higher minimal stent area not only as an absolute value, but as a relative value regarding the reference vessel area. And as higher is this ratio between minimal stent area and reference vessel area, the better are the results. This is one recent study which clearly demonstrated that in left main and in the right coronary, and in general, as higher is the ratio between the minimal stent area and reference vessel area, the better are the results. The lower is stent thrombosis, the lower is target vessel revascularization. So there is a specific requirement in this regard to the stents. And in the left main, there is a higher frequency of calcium, which again makes this region prone to spasm and to stent recoil. Now, when we stent the left main, the pot technique is absolutely obligatory when the distal left main is stented. And the problem with the pot is that it requires really a good stent strata position at the proximal part of the bifurcation, because if you miss something proximally, there is a huge chance of going with the wire behind the stent. And this is the other problem, the axial, the longitudinal deformation of the stent. Actually, the Taxus stent was at the beginning mainly criticized for this deformation, and that was really improved in Synergy and now in Megatron. These deformations lead to worse results, and when the stent is deformed, there is a higher rate in the higher target lesion revascularization rate, which is true for the left main and for the right coronary artery. And the Synergy Megatron was dedicated exactly to cover these needs. It has a highest axial and radial strength. It has practically the highest overexpansion ability at the moment from all stents which we have, at least here in Bulgaria. The scaffolding is, as I said, the radial strength are really excellent, and the stent is visible. This is because of the platinum content, which make it better visible on fluoroscopy, which is, you know, it's really good to see when your stent is, especially for the overlapping segments, when you do pot or if you have to recross the stent struts for side branch wiring, this really a need. The Synergy Megatron has a better radial strength. This is because of the wider peaks. This part from the stent, and practically the radial strength is the ratio between the peak strength and the strut length. These are peaks of the stent, and Megatron has 12 peaks, which make it much more resistant for the vessel recoil. And this is, as I said, this make it much more resistant for the vessel recoil, and this improves the radial strength. The next is comparison with the competitors. As I said at the beginning, the stent Promus stent was exactly the same with Xience. However, they diverge, and as you see, at the moment, Megatron has a 60% more higher radial strength and 40% higher radial strength in comparison with also with the Onyx stent. There is a higher axial strength. That was because of the additional connectors, which are with changed geometry and which are wider, and the axial strength, which make the stent more resistant for the longitudinal deformation. Its axial strength is higher by 44% from Xience and 43% with Onyx. Synergy stent is practically one of the stents with the higher ability for overexpansion, and the overexpansion range is the highest at the moment from all different stents which we have on the market. And probably the only one which is compatible with the Megatron is Onyx, but with the Xience SkyPoint, however, this is true only for the larger diameters. As I said, regarding the visibility of stent, this is because of the platinum content, and it makes stent really well visible. So all these stent characteristics lead to cover the requirements for stenting left main and osteo right, with the maximizing the minimal stent area and avoiding the stent malapposition, and these needs are covered from the Megatron stent. Thank you. Sure.
You're sure aware of the SCAR registry in Sweden. There is a strikingly high restenosis rate about Megatron, also in comparison to Synergy. Do you maybe think about how is this like the mechanism of that? Because they made in left main, maybe because of like the plug difference over there. Let's say I see it all the time, and to compare all this stuff. And
these are data from the
SCAR registry
SCAR.
in Sweden, yeah.
Well, it depends from the technique first. What I learned during the years is sometimes we should be very careful with the technique, and we should follow all the steps no matter what stent you use. And sometimes believing on the advertisement that something is so superior than everything else at the beginning, we are prone to skip some steps from the treating in the best way and following all the technical steps. This is a speculation now.
No, I was also thinking that way because usually the Megatron is implanted in the left main, and how you put it from LED to the left main. And
Exactly.
if you think that, oh, the radial strength is really high, so I don't need to predilate that much, and
Exactly.
maybe because of that, this is the
If you don't
the difference.
exactly the lesion preparation, the pot technique afterwards, just the post dilatation also. These are all necessary steps, no matter what device you use.
Yeah, I also thought
This
so.
is what my personal explanation about that. So, but this is clearly a speculation at the moment.
Of course. Thank you.
Okay. So, thank you. We have a pre-recorded presentation. Zoom. Okay. So.
James Nolan.
Yes, James Nolan will present the data. Can we connect? Regarding these results, I remember how at the beginning of stenting of carotid arteries, I was believing that self-expandable stent placed in carotid has enough radial strength and it will open later on. After several years of stenting, it appeared that this not occurs. Now I'm always do post-dilation with a large balloon. And as I said, this is my personal speculation about this, but probably we should be very, very careful with our technique, how we implant the stents. So, hello, Professor Nolan.
Good morning. Can you hear me?
Yes.
Okay, fantastic. Have you got the slides up on the screen now?
Yes.
Okay, fantastic. So, welcome from a kind of grey and drizzly UK. You won't be surprised to hear that our weather's like that. My name is Jim Nolan. I'm the Professor of Interventional Cardiology in the Cardiovascular Research Group at Keele University in the UK. And I'm going to talk very briefly in about 20 minutes about drug-eluting stent development. And I think my slides won't advance there, guys. I'm not sure what's happening.
We see your screen at the moment, so you can proceed.
Yeah. The slides. Okay. So I want to just briefly remind you of the history of angioplasty and stent development. So in less than 50 years, we've gone from the first balloon angioplasty in 1977, which, you know, these procedures had any of us who did them will know that there were a lot of problems with elastic recoil, abrupt vessel closure, and a lot of patients came back to the lab with recurrent symptoms. Bare metal stents date from 1987, starting with stainless steel slotted tube stent platforms, and while they were in advance, there were significant problems with restenosis, early stent thrombosis, and delivery difficulties. You know, I did use these early stents, and we were regularly losing them. They would disappear off the balloon. You know, we had a lot of problems with them. And the first generation of drug-eluting stents in 1999 were a massive step forward. But unfortunately, the anti-proliferative drug and the polymer were toxic, and that led to problems with late and very late stent thrombosis. And so we've ended up after a near 50-year period with highly effective second-generation drug-eluting stents, which have improvements in stent design and the polymers and drugs to improve their performance and vascular repair. I'm sorry, guys, but okay. What are the factors that drive stent development and innovation? First of all, we know that there were inherent limitations to balloon angioplasty with an early abrupt closure rate of around 1% and a late 30% problem with restenosis. We also are aware that there are compromised mechanical performance for early slotted tube, closed cell stainless steel bare metal stent designs, and the problems were their deliverability and their lack of utility because of their very closed design. And we also had suboptimal drug and polymer performances in the early drug-eluting stents. And as drug-eluting stent design has evolved, PCI practice has evolved away from relatively simple type A, type B lesions into very complex coronary angioplasty procedures. And these procedures need improved delivery, size compatibility, and side branch access features in stents. Now, drug-eluting stents can be developed in two different ways. On the left here, you can see that there are options for platform optimization through changing the metal composition, strut thickness, cell size, and cell strut design to try and reduce restenosis and inflammation and to improve side branch access and vessel scaffolding. And then when we look at the drug and polymer, we need to reduce drug toxicity, enhance biocompatibility, and hence promote healing with a reduced risk of restenosis and stent thrombosis. And that will allow us to optimize adjunctive drug therapy. So the perfect contemporary drug-eluting stent needs to be deliverable so that we can get it into the vessel, conformable so that it does not distort the target vessel geometry too much. It needs to have size and expansion flexibility and be optimized for complex procedures and have an effective and biocompatible polymer and drug. Now, over that near 50-year cycle of innovation and improvement in stents, of drug-eluting stents, there have been many missteps. Here's one paper that looked at the effectiveness of a statin-eluting stent in an animal model where it showed promise, which didn't translate into benefits in the clinical model. And there have been many other false steps and blind alleys with heparin-coated stents, steroid-eluting stents, drugs that didn't work, polymers that were too toxic, etc., etc. So it's not a uniform, linear progression in stent design. There are blind alleys. If we look at stent platform optimization, there have been material developments, and the most important one has been the move from stainless steel into cobalt chromium. And cobalt chromium is stronger than stainless steel, and so stents constructed with cobalt chromium have enhanced radial strength, and they also have increased radio opacity, which means you can construct stents with thinner struts. You can then modify the strut geometry to enhance performance. Now, there are other alloys, platinum alloys, but cobalt chromium is the alloy that's used for most contemporary stents. When you look at developments in stent composition and geometry, this is an original Palmer-Schatz stent. And when you look at it, you can see that this type of slotted tube, it has multiple connections between each cell within the stent itself. Now, what that confers to the stent is excellent radial strength and performance, but it does make it inflexible and it does make it difficult to deliver, and it means that when it's expanded, cell size within the stent is quite small. You can contrast that with this typical contemporary stent, drug-eluting stent, which is an open cell, two-link modular ring cobalt chromium design. And you can see it has very, very different appearance and very different geometry compared to the Palmer-Schatz stent. So the stent is cut into rings, and there are only two connectors between each ring. So this should make for a flexible stent, which is highly deliverable, but maintains its radial performance, whilst also having large cell sizes in its expanded form. And here, you can look at these two pictures. On the left is a contemporary two-link stent, and on the right is a more traditional multi-link stent. And in the expanded form, you can see that the modern stent on the left has a cell size that's at least double that of the older stent. And that is a function of reducing the number of connectors in the design. That is really important in complex angioplasty, and so here in this study, which compared a modern stent in the orange bars and an older stent design in the grey bars, you can see that for all these categories of complex angioplasty, the modern stent outperforms the older stent design. And if you look, I know it's a bifurcation meeting here that we're talking at, and if you look here specifically at the bifurcation procedures, the modern stent was enormously better than an older historical stent. Now, this improved performance with an open cell two-link design is gained without any increase in stent thrombosis or safety. You can see there's no adverse safety signals here. The two-connector design will, along with the thinner struts, is designed to enhance stent flexibility. And you can see here in a lab model that if you look at the new stent compared with an older stent design, that the new stent is significantly more flexible. This is a visual lab model that gives you an idea of what that flexibility means. So on the left here, the new stent is a two-connector modular ring design, and on the right you have a multi-connector, more traditional old-style stent. They're both deployed in identical models. You can see that the new design conforms very well to the target vessel geometry, but here the old stent has problems with opposing at the edges because of its multi-connector design. This is another model study, and here on the left, the new stent is deployed in a bend, and here on the right, an older stent is deployed in the identical model, and you can see that there's minimal disturbance to the target vessel geometry with the new design, only 8% deformity, and here with the older design, you can see that the vessel is much more straight, and there's a 25% change in vessel geometry. So having a flexible, conformable stent has real advantages for the patient. These lower profile, thinner stent struts are much more deliverable. And here, if you look to the right of the slide, you can see that these are measurements made in models with tortuosity, resistance areas, and you can see that the resistance value for a modern design is far superior to the old design. So this stent will go into vessels that these will not enter. A real benefit in routine clinical practice. Coronary vessels are highly variable in diameter, and one size does not fit all vessels. Now, typically, manufacturers have produced small and large vessel stents and workhorse stents. They are all the identical stent crimped onto smaller or larger balloons. But increasingly, we're moving to changes in geometry to reflect the target vessels. So manufacturers will generally can produce a small vessel stent, a workhorse stent, and a large vessel stent. In large vessels, the stent geometry can be advantageously manipulated. So you can have increased strut thickness to provide better scaffolding. You can have extra crowns within the rings that allow more expansion. And you can have increases in cell length and diameter area that should function to improve the stent performance in large vessels. And here you can see a new stent, the green bar, and two old designs, and these are measures of radial force, and a new multi-crown, thicker strut, large vessel stent has superior radial force to these older designs. So these small changes in stent design and geometry do have real benefits in clinical practice. This is a modern 10-crown large vessel stent, 2-connector large vessel stent, and this stent can now be expanded up to 6.25 millimeters. So it's a very large expansile capacity in a dedicated large vessel stent, which is important in dealing with some native coronaries, particularly the treatment of left main stems that can sometimes be in this size range. So modifying your stent geometry for a large vessel stent has real benefits in terms of expansile capacity. When you think about small vessels, simply crimping a workhorse stent onto a smaller balloon is always associated with problems. You can't crimp them to a very small size. Now, a dedicated small vessel stent can be produced which has thinner struts, so you decrease strut thickness, and if you change the strut and band dimensions and shorten them, you will have a thinner strut, more flexible stent that can be crimped onto smaller vessels and should be superior for stenting small vessels. So here we look at the crossing profile of one of these contemporary small vessel stents in green compared with older small vessel stents. First of all, you can crimp the new stents down onto as small as a 2 millimeter balloon, so you've got more flexibility in the size of vessels you can treat. And secondly, the crossing profile of a dedicated small vessel stent compared to older designs is significantly improved, and so the delivery of these dedicated small vessel systems into small vessels will be superior. You can also look at the stent flexibility before expansion and see that the new small vessel stents are superior to the older designs, 14% increase in stent flexibility, so you'll be more likely to get this low profile, highly flexible small vessel stent into those tricky anatomical situations where you need to stent a small vessel. The final area to talk about is polymer and drug optimization. So the first thing is, obviously, it's important to move from a cytotoxic drug like paclitaxel to a cytostatic drug like sirolimus. Cytotoxic drugs damage the cellular environment that the stent is deployed into more than cytostatic drugs. So moving from a toxic to a static drug is important. The next thing is that companies can now deploy stent polymers on the external surface of the stent only in an abluminal distribution. This means that the interior of the stent, where you want endothelialization and healthy cell growth to cover the stent, is not affected by the presence of polymer. Also, bioresorbable coatings are very useful because after a certain period of time, they will be completely gone. And there is some evidence that non-resorbable persistent polymers are associated with inflammation and an increased risk of stent thrombosis. You can also change the composition of the polymers to increase the elasticity, and that is important. So if you look at the two pictures on the right of this slide, you can see what happens to stiffer polymers during stent expansion. They crack and they can displace. Another way, as well as having a more flexible polymer, another way to improve this is to change the distribution of the polymer on the stent. So here, the green areas represent polymer distribution on a contemporary stent. It has a gradient coating so that these high flexion points don't have any or minimal polymer. And so you get less cracking and less potential for polymer-related injury. A bioresorbable polymer will be present for around 120 days, and that will cover these periods of thrombosis, inflammation, proliferation, and at the end of the period of 90 days, which is typical for these phases of of healing and response to an implanted stent, the polymer and its drug will be completely gone, and that will enhance cellular healing. If we look at an optimized drug polymer system, contemporary system, you definitely get very quick strut coverage as early as 1 month. So here in this study, at 1 month with a contemporary system with an abluminal bioresorbable polymer, there was 85% strut coverage at 1 month, and by 3 months, 95%. So it's very quick. Just to show that pictorially here in an animal model, on the left is the older drug-eluting stent in an explanted rabbit iliac artery, and here is the optimized drug-eluting stent. So if you look at this picture, you can see extensive coverage of all the struts in the explanted segment, 97% strut coverage. And here with the older stent design, 73% coverage, you can still see the stent struts. So visually you can see the benefit of having an abluminal optimized drug delivery system. It's not just about the cells, it's about how the cells function. And here the green bars are a contemporary stent compared to the blue bars, which are an older stent design, which is not optimized for drug and polymer performance. So this first study illustrates the number of tight junctions in endothelial cells, which are increased at 7 and 10 days for an optimal system compared to an older system. And tight junctions mean better functioning endothelial cells. So not only do you have more coverage, you have more coverage with better functioning endothelium. And this translates directly into less reduced levels of fibrin deposition with the contemporary stent compared to the older style stent. So less thrombosis and risk of clinically significant stent thrombosis events. And here in this large randomized trial, a contemporary abluminal optimized stent design can be shown to have real patient benefits. So it's possible with these new platforms to stop double antiplatelet therapy in 1 month. And here we look at the risk of an ischemic event for standard, prolonged or abbreviated double antiplatelet therapy. There is no increase in ischemic events. And here we look at bleeding events, and for abbreviated double antiplatelet therapy, there's a significant reduction in bleeding. So these factors of stent design, optimizing the mechanical performance and improving the way the coatings are applied and the way the drugs are delivered, have real benefits for our patients. And when you look here in a very contemporary study, these event rates at 1 year in a 37,000 patient study are negligible, really. It was unimaginable at the start of the coronary intervention era that you would get these sort of results. So when we think about stent design and innovation and how it impacts on the clinical performance of stents, we need to think about strut thickness and composition, because these directly affect deliverability and scaffolding properties, with cobalt, chromium or related alloys representing a significant step forward. We then need to think about strut configuration, and contemporary modular ring designs with reduced connectors aids flexibility in delivery without compromise to scaffolding. The connectors being reduced in number is very important to generate an open cell design, because this will improve side branch access, which is particularly important in bifurcation interventional procedures. Then we need to think about the delivery polymer and its compatibility, whether it's distributed abluminally and whether it's biodegradable. And there's clear evidence that supports the design of abluminal bioresorbable drug polymers with enhanced flexibility and a cytostatic, not a cytotoxic drug. So, in conclusion, it's a short talk, but I hope you can see that drug-eluting stent development programs aim to produce optimized contemporary drug-eluting stents with favorable cell geometry for complex angioplasty, with large overexpansion capacity, and by modifications, we can extend the therapeutic window into both very large and very small vessels. Drug and polymer design importantly enhances healing. You can see that the stents are largely covered after 1 month with contemporary systems, and that's very important because it supports tailored double antiplatelet therapy in high bleeding risk patients, and a lot of our patients have high bleeding risk features. And the design features of modern stents are reflected in very favorable clinical, safety, and performance data. Thank you.
Thank you, Professor Nolan. Can we keep connected?
Yeah, sure, yeah.
Okay, because the screen was changed. That was really educational presentation. I think that it will be really useful for our audience here. And now it's open for questions, comments from the panel. Ravi?
Yeah, I had a comment and maybe a question also. So we have polymers specifically, because I think we know what all the Olamus drug have so far worked pretty well. I have a little bit of a puzzling situation where all of the polymers are now somewhat biostatic in the sense they produce a very low level of inflammation. So, and in clinical trials, I'm not seeing much difference in the stents that have biodegradable polymers versus the polymers that are non-biodegradable, because they're both biostatic. When you look at Science and Synergy data, including the short DAPT trials with both of them, as well as the TLR and other MACE over years, they're pretty much the same. So theoretically, the biodegradable polymers should have a distinct advantage, but in clinical trials, I don't see that much. I think it is good to have biodegradables, so less of the stent material, except for the scaffold, except for, yes, for the cobalt chromium scaffold, everything else is gone. It seems beneficial, but I think in clinical trials, I think the stents and the drugs are so good that you just don't see much of a difference. Is that pretty much a correct observation?
I think that's completely correct, yeah. I mean, you can see very little difference between contemporary stent platforms in most cases, most of the time. It's perhaps when you look at, small, higher risk subgroups that you see the benefits of the small nuances and gains of optimized stent designs and polymers and drugs and things like that. You can see differences when you look in animal models or when you look at bench testing, but I think it's fair to say that the systems are so good across degradable, non-degradable, different drugs. They are so good that it's hard to see significant benefits in clinical practice.
I agree, yes, I think.
What are the data from the SCAR? Because
the SCAR data,
okay.
the stents that have abluminal, let's say Synergy and Ultimaster, have in stent thrombosis area more stent thrombosis than Orsiro and Onyx that have basically the polymer everywhere. This is the,
Biodegradable polymers?
Yeah.
More stent thrombosis?
No, so Orsiro and Onyx in the SCAR registry have less stent thrombosis than those who have just the abluminal polymer. It's a Synergy and Ultimaster. Okay. Yeah, it's just a matter of, because it was made like, okay, it's less stent thrombosis because of no polymer in that area. This real world data, actually it's somehow then intriguing and the opposite, and it shows that Ultimaster and Synergy have less restenosis actually than these actual two stents, Onyx and.
What's the mechanism?
I don't know. I'm a clinician. I just look at the data.
Yeah. Is that the data from the SCAR registry?
Yeah.
I mean, it's difficult to know, isn't it, from registry data, because it's perfectly feasible that, confounding factors drive those kind of differences in a registry. A lot of my academic work has been around registry data, and there are things that you can see, and there are also unmeasured confounding. So, in a registry, if one particular stent is used in more of the complex patients, that alone could drive the difference. There are things in registries that are measured and unmeasured that can account for kind of differences.
Yeah, but well, the SCAR registry is like all implanted stents. I mean, that's a good thing about Sweden, because then you can see that they follow basically every patient, and also they have the chart about which stent is implanted the most. So Synergy is implanted the most, Onyx a lot, and then Orsiro a lot, Science still a lot. So I always like to look at their data, because they are like real life. There are no exclusion criteria.
Exactly, yeah. So, registries are really useful for that, aren't they? I mean, in the UK, we have the BESIS registry, and that's generated a lot of data around radial access and these kind of things. But, there's always a caution with registry data.
Yeah. No, we were speaking before. Maybe you know that in the stent restenosis rate, the Megatron has a way higher restenosis rate in that SCAR registry than the Synergy. And we were speaking that the technique, the implantation technique, maybe if you know that the radial force is higher, and I put just a stent and I don't need to post-dilate or pre-dilate or things like that. So it encompasses also the stent implantation techniques, not just the stent basically per se, how it is and his, let's say, abilities or strengths.
Yeah. It's interesting, because I would never implant a stent without pre-dilating the lesion. Never.
Hi, Jim, it's Lambros. First of all, my greetings to
Stoke. Lambros, hi.
How are you?
I'm good, thank you.
Great. Just a simple question. Nowadays, stents are very good. Do you think there's a room for improvement? And do you think there's appetite for investment? And if so, towards what is going to be the focus?
I think it's really hard to get significant incremental improvements over and above where current cobalt chromium, modular ring, open cell, optimized polymer stents with a good drug. I think it's really hard to improve that. So I think you'll see that there'll be a period of relative stability. I mean, the amount of effort that has to go into generating a small change in a stent is enormous. So, I know that producing that gradient coating, for instance, on the external surface of the stents that spares the flex areas was incredibly difficult to do, and it took years of work to get that right. So I think all of the systems are excellent now. You can largely choose what you want if you choose to, you look at them. I personally would look for a 2-connector open cell, cobalt chromium thin straw abluminal design, but there are a number of them. But I think it's going to be, I don't think you're going to see massive innovations in stent design and delivery. I mean, I started off using hand-crimped Palmer-Schatz stents. They used to be, to get a short stent in those days, we used to cut them in half with a pair of scissors on the table and then implant the little half of a stent. I mean, we've come an enormous way, but I think we're probably into a period of relatively static stent innovation now.
Thanks. I agree, but just I want to listen to your view as well.
Yeah.
Okay, so thank you so much. It was a really nice discussion, and we have to proceed because we are out of time. And thank you, Professor Nolan. So now there is a time for some fresh look about the new type of drug-eluting stent. And Dr. Foros.
Hello, everybody. Dear committee, dear presidents, dear professors, thank you for inviting me here to speak about maybe a new idea beyond the Limus family. Is there any place for something new, maybe for a new retinoic eluting stent in the cath lab? nowadays, cardiovascular disease still is the leading cause of death worldwide. And nowadays, we have a very good tool, a supportive tool, the new generation, second and in specific third generation stents, which are the gold standard as safe tool for acute and chronic coronary syndromes, PCI. What was the evolution from the first balloon angioplasty in 1977 from Dr. Grunzig? Because of the abrupt closure and elastic cold restenosis, we found the solution first of the bare metal stents. Because of the restenosis and stent thrombosis, then came the first generation drug-eluting stents, which had but the problem of the high stent thrombosis. Then came the second generation stents, which were related with late and very late stent thrombosis. The permanent implant state and was really difficult sometimes to come to challenging lesions. Then the third generation stents solved some of the problem, but the stent remained there. Drug-coated balloons and bioresorbable scaffolds came as a solution for these problems, but they were also having some problems. I want a little bit to say for the three main components of the drug-eluting stents. We have the polymer coating, the stent platform, and the anti-proliferative drug. Much evolution in all three things, and particularly for the anti-proliferate drug, we are today in the Limus family. Paclitaxel in the first Taxus stent was used, but nowadays we don't see that so often or anymore, and actinomycin D, which was used, is not currently in use anymore. What are the problems mainly for the new, that remain in the new generation, second and third generation stent? The main problem is the in-stent restenosis, the so-called ISR. It's rare, but it's rare, but it remains a major problem when somebody sees how many people are nowadays treated with PCI, and sometimes it's a fatal complication. In some registries and observational studies, we see an amount for 10% ISR rates, and according to the nowadays research, the 5 or 10% of all percutaneous coronary interventions are performed in ISR, which are the most, one of the most challenging lesions today. The main mechanism of in-stent restenosis are the neointimal hyperplasia and the continuous inflammation. So, what are the disadvantages and the complications which are related to the new generation stents? They have to do with currently used ketostatic drugs, which have to do with local inflammation. They delay or prevent the reinthalization. They can sometimes cause arterial cell toxicity, and they require dual antiplatelet therapy. So, today, after the whole evolution from 1977 to today, we have a very good tool in order to treat our patients in a very good way, but there is still place for a new, maybe metal design, maybe new polymers, new drugs, which are investigated. So, I think, and we all should think so, that there is time maybe for something new. And that's what we research in our university in Ioannina. If there is place maybe for a retinoic acid eluting stent or a retinoic acid coated balloon. We will see what we found in our university. Some few words for retinoids. They are a class of natural and synthetic compounds with chemical structure similar to that of retinol, vitamin A. They include carotenoids, retinol, retinyl esters, retinal and retinoic acids. And retinol is physiologically converted to all-trans retinal and then to all-trans retinoic acid. Then ATRA binds to specific nuclear receptors to perform its biological action and inhibits cell proliferation and induces differentiation in various cell systems. That's a schema, a figure how this works, how this binds and then comes in the cell, in the nucleus of the cell. So, especially how do the retinoids act in the cardiovascular system? They are essential regulators of the cardiovascular system in the manner of cardiac function, cardiac regeneration, and cardiovascular disease progression. They are necessary for normal embryonal heart development, and they play a significant role in the apoptotic pathways and death in experimental models of acute myocardial infarction. In contrast, oral administration of ATRA-induced cardiac hypertrophy in a dose-dependent manner in normal rats. In atherosclerosis, there are some experimental models that examined the role of retinoic acid administration in the development of atheromatous plaques and restenosis. And the main results from these models shows that retinoic acid mitigates the development of plaque burden in arterial tree. Also, retinoic acid receptors seems to regulate various steps in cell differentiation, inflammatory cell adhesion, and activity in the process of atherosclerosis. These are the experimental models which investigate the systemic delivery of retinoic acids and experimental models which served a local delivery of retinoic acid. What are the limitations of retinoic acid in clinical practice? Unfortunately, the dose of oral administration of ATRA used in experiments is impossible to be used in clinical practice because it brings the so-called ITRA syndrome with large complications as liver dysfunction, pulmonary infiltration, and can be fatal in 1 or 2% of cases. Also, there's no, in the data of nowadays, there is no available information on the long-term effects of local delivery of RA. We have, in the most surveys, an observation period of 4 until to 12 weeks. The smooth muscle cells cultured from other experimental models retain a higher degree of differentiation than that described in studies extracted from rabbits and rats. Also, there are no investigations in coronary arteries, but all the surveys are in non-coronary arteries of laboratory animals. So, the survey, the firms, the investigators tried to make better the stent, the drug-eluting stents by trying to leave nothing behind. Promising were the scaffolds, but some years ago, the Absorb 3 and 5 studies made them rule them out from the coronary labor. As the speaker before said, the new scaffolds are trying to come again in the practicing of PCI, and we will see what the future will bring. So, concluding all what I've said until now, I would like to show why is it time for retinoic acid eluting stent to come. Retinoic acid could be less cytotoxic or cytostatic. Retinoid acid signaling plays a role in the atherosclerotic progress in manners of reduction of neointimal hyperplasia, stenosis, or restenosis rate after injury, graft, or stent placement in non-coronary arteries of experimental models. And we know that restenosis is still a major complication in the drug-eluting stent era. Chronic inflammatory response and a need for antiplatelet, dual antiplatelet therapy. That's why I think, and we all think in our university, that further investigation and development of technologies is still needed. What have we seen? We evaluated histopathologically a retinoic acid-eluting stent in a rabbit iliac artery model. We used cobalt chromium stents coated with bioabsorbable polymers and all trans retinoic acid from the firma Rontis. We had retinoic acid local delivered in the iliac artery, and our experimental model was rabbit iliac artery. Our methods, we used 60 stents in 30 rabbits, male New Zealand, in 10 groups divided, 2 polymers, A and B, 3 doses of retinoic acid, low, medium, and high, in 4 control groups with 6 stents per group and 3 animals per group. That's how we divided our models with the two polymer systems in low, medium, and high dose, and we had a bare metal stent as reference item 1, and the Xines everolimus Dragalume stent as reference item number 2. We implanted the stent in iliac arteries of rabbits, right and left. We made OCT in 4 of our models with this picture, and we explanted in 4 weeks, and then we had a histopathologic evaluation. We evaluated the arterial reaction, the artery inflammation, the medial smooth muscle cell loss, the artery cell replacement tissue, the host reaction associated with the implant, the elastic lumina rupture, and the medial hypertrophy. And we made the quantitative evaluation by image histopathological analysis. So, what were our results? We found in this group of rabbits that in all groups no thrombus formation or necrosis was found in the vascular wall, no dissection of the wall, only multifocal ruptures of the elastic laminae. We found low host reaction with all the treatments, and unstained particles within the tunica intima in association with macrophage and giant cells, except from our group C. We found that total arterial reaction scoring had a difference of 5.2, that is low, and we found lower total arterial reaction in the groups from the second polymer and the groups from drug-coated balloon versus the bare metal stent. So what we ended up is that there's no statistically significant difference between the groups of the retinoic acid group in comparison with the bare metal stent and the group with the group of the bare metal stent and the group of the Xines everolimus stent in medial area, intimal area, stenosis, intimal mean thickness. That's what we found in more details. That is in intimal median thickness there's statistically different significance different in versus the group A and the group B, and versus in group A versus group A8 and versus the bare metal stent. In terms of the stenosis, the first group with the retinoic acid showed the highest stenosis. And in terms of the intimal area, we found statistically significant difference between groups between the group of the drug-coated balloon and the bare metal stent. So, in conclusion, in order not to confuse with you with the whole histopathological results, is our observation showed that none of the treatments seems to increase the risk of a thrombotic event. There is a low risk of arterial transmural dissection induced by the stent implantation. We found that the host reaction associated with the implant struts was considered low with all the treatments, and that the dose of retinoid acid and the type of the polymer may play a role in the biocompatibility of the device. Naturally, there are also limitations, as in every experimental model. All stents were implanted in healthy iliac arteries. The iliac arteries are elastic natured and are naturally different from the muscular coronary arteries, and the observation period was only 4 weeks. I think that now it's the time to investigate the local delivery of retinoic acid in peak coronary artery, what we have made, and we will have the first results in the next months. We hope that in the future we will make the first experiment in coronary arteries of a peak model, and if then the results are promising, a first-man study could be designed. Concluding, what I want to say with this presentation is that we have naturally a very good tool, a gold standard tool in our cath lab for treating our patient, drug-eluting stents, but we should always try to make it better, or to find solution in the problems that the drug-eluting stent had brought. Thank you very much.
Thank you so much. That was a fresh look and new approach, relatively new approach. And Fari?
That's very interesting. I really appreciate it. Fantastic presentation, because the new things, we are used to our stents and we are okay with our stents. And when we saw something interesting, that maybe we change. My question is how the delivery. Thank you very much. How you deliver the retinoic acid over the stent, and first of all, how you obtain retinoic acid? I mean, from the olives probably, because Greece is rich in olives. Probably you finished the olive oil there, and how you deliver it over the stent with a polymer, without polymer.
We made it with polymer. As you saw, we had 2 types of polymer, A and B, in order to see if the polymer plays a role. And we found that polymer can sometimes play a role, but what we saw is that what plays naturally a role is the amount of the retinoic acid what we give. It's a survey. We have to wait. We don't know how will it come in future. But what I would like to say, what I would like to bring you here, is that we should always investigate. We should not stay in what we have. We should always try to make our tools better.
Thank you.
I have a question, because looking at your data, I saw that practically it was not statistically significant, but of course for the animal experiments it's very difficult to obtain statistical difference, but there was a numerical difference between groups with low and high retinoic acid concentration, and practically group with high retinoic acid concentrations had the highest intimal hyperplasia. The intimal thickness was higher, and the percentage diameter stenosis was higher than the low concentration. Did you have some explanation? Because in theory it should be that high retinoic acid concentration should be more cytotoxic or cytostatic, and the results which you present are practically demonstrating reverse, that low concentration of retinoic acid demonstrates better results.
We assume, but thank you firstly for your question. We assume that maybe a local delivery of retinoic acid has not the same effect as we thought that it would be. Maybe the local inflammation that makes is not the one that we expected that would have, and that's why I think that more survey is needed in this.
Yes. Okay. Thank you. So, another comments? If not, thank you so much.